In this video, we will discuss about how this isoferulic acid suppress NF-kappa beta pathway in the pancreatic cancer cells. So far, we have understood that isoferulic acid will cause suppression of proliferation and metastasis in the pancreatic cancer cell lines, and we have tested the evidence in PANC1 cell and BXPC3 cells. Now they wanted to check which pathways get uh, dysregulated by isoferulic acid. Since NF-kappa beta pathway is related to the cell survival assay, so they decided to check with this NF-kappa beta pathway. So once they check some of the proteins that are upregulated during this NF-kappa beta pathways in different conditions of isoferulic acid treatment, and they are able to see that compared to the control, there is a decrease in the uh, proteins or factors that gets upregulated when NF-kappa beta pathways are being triggered. For example, phosphorylation of IK beta alpha and IK beta phosphorylation of IK beta alpha gets downregulated, whereas IK beta alpha gets upregulated. So when the NF-kappa beta pathway is activated, it will get phosphorylated, but this phosphorylation of IK beta alpha gets suppressed whenever the cells get treated with isoferulic acid, indicating that the isoferulic acid suppressed the NF-kappa beta pathway that could cause less survivals of the pancreatic cancer cell lines. And also they check these P65 levels, which is also a factors one of the factors for NF-kappa beta pathway, and they can see that in the case of control, the amount of P65s are higher, but when they are treated with the isoferulic acid, the P65s proteins levels goes down with increase in the concentration of isoferulic acid. That is, for the isoferulic, for example, the isoferulic acid at highest concentration, the red signals goes down significantly compared to the control. And same things they have observed for the BXPC3 cell lines. Here also they see the decrease in the phosphorylation of NF-kappa beta and a decrease in the P65 levels with increase in the concentration of isoferulic acid treated with the increase in the concentration of isoferulic acid to the cells. For example, this is the control P65 signals, and these are the signals for the P65 in cells that are treated with high concentration of isoferulic acid or 12.5 macromolar concentration of isoferulic acid. Now, in order to confirm it's the NF-kappa beta pathway that cause suppress of the that cause decrease in the proliferation of migration and metastasis, what it is, they artificially activate the NF-kappa beta pathway in the isoferulic acid treated cells using NF-kappa beta activators and check whether it will helps in regaining its survival and metastatic properties or not. So first they did a tunnel assay, and for the control we can see that the less amount of cells are apoptotic, but for the isoferulic acid treated cells, we can see the more signals for the apoptotic cells for the tunnel assay. But when they are also treated with NF-kappa beta activators, there is a decrease in the apoptotic signals, indicating that the NF-kappa beta pathways with the activations of NF-kappa beta pathways, the proliferations and migration properties can be restored in the pancreatic cancer cell lines. And similar results they have observed for these BXPC3 cell lines. That is, for the control they see less tunnel signals or apoptotic cell signals, but for the isoferulic acid treated cells at high concentration they see more apoptotic cells. But when these cells with high isoferulic acid concentrations are treated with NF-kappa beta activators, they see a decrease in the signal for the apoptotic cells, indicating that NF-kappa beta pathway activators leads to the activation of NF-kappa beta pathway. It will and whose function is to help in the survival of the cells. And they also checked these apoptotic factors in different cell lines, apoptotic factors in different conditions of the isoferulic acid, and they can see that when the cells are treated with high concentration of isoferulic acid with NF-kappa beta pathway, the apoptotic factors goes down compared to the case where cells are only treated with isoferulic acid at high concentrations. And this is true for PANC1 cells and also for this BXPC3 cells, we can see that cleaved PARP goes down when the cells are treated with isoferulic acid at high concentration plus NF-kappa beta activators. But when these cells are not treated with NF-kappa beta activators, we see that high cleaved PARP or apoptotic factors. And the P65 signals are also shown here when the NF-kappa beta activators is used, and we can see that from compared to the control case, when only the isoferulic acids are used at high concentration, the P65 level goes down. But when this NF-kappa beta pathway activators are also used, they see that P65 level again goes up. And this is true for PANC1 cells and also for these BXPC3 cells. That is, BXPC3 cells in control, compared to the control signal, the P65 level goes down when the cells are only treated with isoferulic acid. But when they are also treated with NF-kappa beta activators, they see an increase in the signal for this P65, indicating that NF-kappa beta activation or NF-kappa beta pathway will cause will trigger survival of the pancreatic cancer cell lines, survival and proliferations of the pancreatic cancer cell lines, as this is one of the functions for the NF-kappa beta pathway. So I hope you understood how this isoferulic acid is related to the NF-kappa beta pathway, or how it suppresses the NF-kappa beta pathway, which in turn leads to the suppression of proliferation and migration properties of the pancreatic cancer cell lines. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.